moving up to the macaque monkey, to the human, that is tenfold larger in surface area. Just a quick reminder that the cortical turf that you have is about 1,000 square centimeters per hemisphere, and just to make it tangible, that's about a 13-inch pizza on each side. And as a crude first-order approximation, each of the cortical areas we want to understand is, on average, about a pepperoni's worth. But, as with macaque, there's tremendous variability, and I think that's best exemplified at this stage by taking two sets of cortical areas that are pretty well understood and have been mapped to our PALS atlas. So if we take the visuotopic areas done from various fMRI studies on one side, and the orbital frontal areas analyzed architectonically by Joel Price, we find that on average they're about 500 millimeters squared. There is a 100-fold range in size in humans as in macaques, but if we take that average, we can estimate there's going to be somewhere between 150 and 200 cortical areas if they fall within this range. And again, that's based on the assumption that may be challenged, that the entire cortical sheet follows a mosaic organization as distinct from gradients of connectivity and function that may not have sharp borders everywhere. Expanding on points I made earlier for the macaque and touched upon already, every brain is unique, and the kinds of individual variability we need to acknowledge and wrestle with start with just the macroscopic differences in size and shape. Those are fairly straightforward to compensate for. Much harder is the tremendous variability in cortical folding patterns, plus the fact that in humans, as in monkeys, individual areas vary twofold or more in surface area, and there are prominent variability in the relationship of cortical areas to the underlying cortical folds. And lastly, the wiring of every person's brain in this room is going to be dramatically different in ways that account for our different cognitive and personality characteristics and other aspects. I'd like to touch briefly on the relationship between monkey and human. The monkey macaque as a model system is going to continue to be very informative in interpreting what we learn about human function and connectivity, but we have to recognize that the relationships are complex and very nonlinear. Basically, the human cerebral cortex during recent evolution has expanded tremendously in surface area, we know that, but also very non-uniformly, as illustrated by this map of hotspots of evolution that are concentrated in lateral, temporal, parietal, and frontal cortex. And so that's going to make it an interesting challenge to relate across the species. So finally, to summarize, partitioning cerebral cortex is a really hard problem to this day. It's absolutely critical to take us to the next level of understanding brain function. When we have reliably identifiable areas in individual subjects, it'll be possible to intensively study their function and organization in very informative ways. The analysis of connections and the nascent field of human connectomics poses even greater challenges. We have to recognize it, but it's equally critical. It's circuitry that drives our functions of the brain and our behavior. If we think at one level, we can say, well, gee, if we could just get a wiring diagram that spanned a couple hundred areas and maybe counting subcortical 40 or so connections per area, there's a typo, that's 8,000 pathways to get a handle on. That would be fabulous, to be sure, but let's never forget that the human connectome is really, must entail an understanding of how billions of neurons make thousands of synapses and give us an offset petabyte worth of connections. So thank you for your time, and we look forward to hearing where the competition has taken us.